Okay, welcome back everyone. This next video looks at genomes and mutations. Now, of course, we know that the genome itself is all the genetic information encoded by a complete set of an organism's chromosomes. Genes code for protein, of course, but in eukaryotes, only a small fraction of that, the entire genome is made up of genes. The remaining DNA does not code for protein. Now, if we look at these non-protein coding sequences, they're involved in the regulation of transcription and protection of chromosomes. In terms of regulation of transcription, RNA polymerase, the enzyme which transcribes a primary transcript of mRNA from the DNA template, that can't do it all alone. It needs to have transcription factors to assist in that. In terms of protection, telomeres, which are repeating sections of non-coding DNA, are found at the ends of the chromosomes, like so. And every time a cell divides, when these chromosomes are pulled apart, a tiny bit of those ends fray off, and essentially telomeres are associated with the aging process. If you have a look a bit more about those non-translated forms of RNA, that can be transcribed from DNA. Of course, we've seen already tRNA, transfer RNA, which brings specific amino acids to the growing polypeptide chain during protein synthesis at the ribosomes. And ribosomal RNA, incorporated with protein, provides a very important element in functioning ribosomes, and of course is responsible for forming the peptide bond between amino acids as the polypeptide grows. So if we carry on and continue to look at mutations themselves, of course, a mutation is a change in the structure of an organism's genome. It can be as little as a change in a single base in the base sequence, or it can be a very major change in terms of chromosome structure or number. Any mutation may alter the protein being produced, or may result in no protein at all being produced. And any such organism that possesses a mutation is, of course, a mutant. In terms of the frequency, mutations are random, they occur spontaneously, and they're generally quite rare, but they can be increased, or the rate of mutations can be increased by various mutagenic agents. So these would be chemicals, for example, such as mustard gas or radiation, gamma rays, X-rays, or UV light. So if we begin to look at the different types of mutations themselves, if we start with point mutations, which are basically changes in a single base pair in the DNA for a single gene. Now that could either result in a change in the protein coding region or the part of the gene that regulates the transcription of that protein coding region. There are three types of point mutations: substitution, insertion, or deletion. And in each case, as we'll see, it's the codons for the amino acids which are altered as a result. In terms of impact, substitution point mutations are where one base is swapped for another, so it changes only one amino acid in the entire protein structure. However, insertion or deletion point mutations, as the name suggests, is where an extra base is inserted or a base is taken away, and all the codons after the mutation will be affected as a result, so they could result in major changes of the protein structure or its function. An example of a substitution point mutation, sickle cell anemia, where a single change in the base results in the substitution of glutamic acid for valine, and that results in abnormal hemoglobin found in those sickle cells. Now, how that works is, if we have normal hemoglobin, here's the DNA strand for that. The RNA strand transcribed from that will be like so. And the parts of the protein, each three bases, of course, representing one codon. So in this instance, we've got enough here for three amino acids: proline, glutamic acid, and glutamic acid again. However, in the sickle cell hemoglobin, there's the DNA strand. However, this thymine here is replaced with an adenine instead, and that ends up with the RNA strand with the codon for the amino acid being altered from GAG to GUG, and that codon codes for valine rather than glutamic acid, and that's responsible for the change in the shape of the hemoglobin molecule. There are several types of substitution point mutation. This one is what's called a silent point mutation, where this mutation does not result in a change in the amino acid incorporated into the polypeptide. So here we see the actual substitution that's taken place, but that codon still codes for lysine. Okay, so there's no actual change. The next one, neutral, which results in a different amino acid being incorporated into the polypeptide. However, the actual chemical properties of the new amino acid are very similar to what was there originally. So originally the lysine, but a change in the codon results in arginine being included, which is chemically very similar to lysine. So there's no actual change in the function of the protein. The next one is called a missense point mutation, where lysine now is altered. The lysine incorporated into the polypeptide chain is now exchanged for glutamic acid, and there's the point mutation that brings about that change, glutamic acid being chemically different to lysine, and so the protein that results is now chemically different and has different properties and will not function as normal. The final one is called a nonsense point mutation, where the resulting change, the resulting point mutation, brings about a stop codon, and so the protein is not produced beyond that point. So of course it's non-functioning. Now of course we've already mentioned insertion and deletion point mutations, where a base is either inserted or deleted from the base sequence, and that results in all the codons thereafter being altered. So of course at that point the resulting protein is most likely to be non-functioning, and because all the codons are altered, these are called frame shift mutations. If we have a look at a bit more detail of how an insertion point mutation works, there's a DNA strand yet again with its base sequence. There's the resulting RNA strand that is transcribed from that, and that results in this sequence of amino acids in the polypeptide. However, in the insertion, guanine is inserted in between the guanine and thymine on the original strand, and that results in a change in the RNA strand, and all the subsequent codons for the amino acids are changed thereafter, which results in all the amino acids being changed after that. For deletion, same principle. There's the original DNA strand and RNA strand again, and the original sequence of amino acids in the protein. However, in this instance we now have a DNA strand, at which point the thymine will be deleted like so, and when the ends of the DNA strand rejoin, all the codons are shifted and all the amino acids thereafter are changed as well. Okay, so insertion and deletion are known as frame shift mutations. Another mutation, either a substitution or insertion or deletion, but an example of a point mutation is a splice site mutation, where the splice sites between the exons and the introns is altered somehow by a point mutation, and this can result in some of the introns being retained or some exons not being included in the mature transcript. And an example of that, a disease from that, could be another example of abnormal hemoglobin, this time in a condition called thalassemia, which results in malformed red blood cells. Okay, so in terms of the evolutionary importance of mutations, most mutations are quite bad. They result in inferior phenotypes that can be selected against. But without mutations, there wouldn't be any genetic variation. All our genes would be homozygous for all organisms. But some mutations can offer some kind of benefit to the organism. So these mutant varieties, as it were, are selected for by natural selection. And of course, natural selection is the driving force of evolution. So finally, we have a look at chromosome structure mutations. These are generally far more serious than point mutations because it involves some kind of breakage occurring in the entire chromosome structure. And they're often quite lethal, but they can happen in several ways. The first one being deletion, as the name suggests. There's a length of DNA. Each of those colored blocks represents a gene. The chromosome breaks in two places, and the segment becomes detached, and then those genes are lost. Okay, the ends of the chromosome join up together, so we end up with a shortened chromosome, as it were. An example of that being Cri-du-Chat syndrome in chromosome number 5, and there's the actual deletion occurred there at the end of the chromosome. The next type, duplication, which occurs in homologous chromosomes, chromosomes that match up with each other, and you can see that by the sequence of genes, which is the same in each of the chromosomes. Essentially, the break occurs in both of those chromosomes. Some of the genes are swapped over. However, in one of those chromosomes now, the genes are duplicated. Okay? This can be bad in many instances. Can be a common cause of cancer. However, there's a good example of a fish that lives in the Antarctic where the duplication has resulted in its ability to produce a glycoprotein which acts like an antifreeze. Next type, an inversion. Once again, breaks occur in the chromosomes and the section of genes flips around and is now back to front, as it were. There's the reverse section. Okay, so after the genes have been inverted within the chromosome, when that chromosome tries to pair up with its non-mutated homologous partner, it can't do so properly. So often non-viable gametes are produced during meiosis. Now the final type, translocation. Now this is swapping of genes between chromosomes that are not the homologous partners. Now in the first instance, a section of genes moves from one chromosome to another. That's called non-reciprocal translocation. And the second is where both chromosomes swap a certain number of genes, like so, and that is reciprocal translocation. Once again, because of the altering of the sequence and number of genes, it results in non-viable gamete formation during meiosis. So finally, if we have a look at those different types of mutation as a summary, in terms of point mutations, we have substitution point mutations, of which there's silent, neutral, missense, and nonsense. And we also have insertion and deletion, which themselves are frame shift mutations, altering all the codons in the RNA strand thereafter. Chromosome mutations, as we have just seen, deletion, duplication, inversion, and of course translocation, of which there is reciprocal and non-reciprocal. Thank you very much.